Hi everyone. In this video we're going to be talking about both cellular and humoral immunity, and so I thought what we would do is start by talking about T cells or T lymphocytes and discuss sort of their origin and preparation. So originally T cells are formed in the bone marrow, red marrow of the bones, and we begin with these hematopoietic stem cells, which are also referred to as hemocytoblasts. And what we're not going to do is discuss the other cells that are formed beyond the lymphocytes, but it is important to note that these stem cells will give rise to ultimately all of the different types of blood cells, including red blood cells, white blood cells, and platelets. So the hematopoietic stem cells can differentiate into lymphoid stem cells and myeloid stem cells, and it's lymphoid stem cells that we're going to focus on here. So lymphoid stem cells, in the case of T cell formation, will be transferred to the thymus for further development and maturation. And once the cells are sort of fully grown in the thymus, the T cells have to undergo a little bit of testing to ensure that they're safe for the body. So there are two parts to this test, and ultimately we just want to sort of help the cells survive that pass a test, and then annihilate the cells that don't pass the test so that they don't hurt the body in some way. So first we're going to test for immunocompetence, which means that we want to make sure that that the T cell can recognize a self-MHC. So you have a body cell that's displaying an MHC with an antigen on it. And in this case, it's going to be a self-antigen. But what's really important is that this T cell is able to recognize the MHC as something that belongs to itself. So if it recognizes the MHC, then it can move on and we can test for tolerance or self-tolerance. And in this case, we want to make sure that the T cell is not going to destroy the self-antigen. So it needs to recognize it as a self-antigen. And then in those cases, what normally would occur in the body is that it would just sort of float on by. So if the T cell fails either of the two tests, then it will be destroyed. But if it passes both, it will then be transferred to the secondary lymphoid organs like the spleen and the lymph nodes. And that's generally where it's going to encounter antigens. So they're just going to probably hang out, wait for antigens to show up, and then when it meets the antigen that it's supposed to meet, it will confer the appropriate immune response. All right, so let's move on and talk about cellular immunity. And so basically what we're going to do is talk about the different roles of the T cells in cellular immunity. And cellular immunity is equally called cell-mediated immunity, so think of those two as interchangeable. So let's draw a nucleated body cell with a class 1 MHC protein displaying a foreign antigen. And then we have our T cell. In this case, it'll be a CD8 cell that's going to sort of be waiting for this cell with the foreign antigen to float on by. And so let me just offer a point of clarification. CD is cluster of differentiation, and it's basically just a numerical coding system for classifying proteins on white blood cells. And so CD8 cells will become cytotoxic T cells or killer T cells. All right, so this CD8 cell is going to encounter foreign antigen, and it wants to confirm that this is definitely a foreign antigen with this process called co-stimulation. So there's going to be like another CD8 protein that will sort of reach out and just sort of confirm by communicating with the cell and the foreign antigen that it is in fact definitely something that needs to be eliminated. And we can also have CD4 or helper T cell involvement. So CD4 would be the immature version of helper T cells, and we're going to explain those next. We can also have helper T cell involvement in this process, and there's a variety of things that can be done, but among them, especially if this is an antigen presenting cell as well as a nucleated body cell. So I don't want to imply that just because it's an APC it can't have MHC class 1. The most common example would be dendritic cells, and dendritic cells they can actually display both class 1 and class 2 MHC proteins. All right, so what we're going to get next is T cell activation. And T cell activation means that this particular T cell is going to be dividing into a couple of different cells. One is the TC cells, or the cytotoxic T cells, and they're essentially just sort of clones of the original cell. And then we can also make memory T cells. The memory T cells are not active, but down the road, if this particular foreign antigen was to re-enter the body after it's been eliminated, then the body would already have these memory T cells just sort of waiting, and the immune response can be a lot faster rather than having to go through all of these processes. All right, so once we have both our memory T cells and cytotoxic T cells, the cytotoxic T cells will attack all of the infected cells. So it could be assumed that if one particular cell possesses a foreign antigen, other cells would possess the same foreign antigen, and it's up to these cytotoxic T cells to go and find them. So cytotoxic T cell is going to encounter the infected cells, and there are a couple of different things that it can do. But most commonly, we will just discuss one example. They could release perforins and granzymes by exocytosis. So similar to the natural killer cells, the perforins will create pores in the cell, which obviously creates free flow of water and stuff into and out of the cells. And then also the granzymes can sneak in through those pores and cause destruction of the cellular component. So ultimately, the cell is going to burst and die. But we don't want to have any of these infected cells circulating throughout the body. Another thing that's going to happen is, and this is a much slower response, but the CD8 cells can also differentiate into suppressor T cells. And the suppressor T cells, they sort of come later, and they inhibit the functions of the cytotoxic T cells in that whole process. And that's really just to sort of keep the system in check and make sure that it doesn't provide an excessive and unnecessary response. Next, I want to talk a little bit about the CD4 cells and their process of activation. So CD4 cells are going to interact with class 2 MHC only. So we have an antigen-presenting cell, and we have our class 2 MHC, and then we have our foreign antigen that is present. And then we have our CD4 cell, which is just in another type of T cell that can only become a helper T cell. And that particular cell is going to come by and recognize this foreign antigen on the class 2 MHC. And once this recognition occurs, and sort of similar to the process that we described for the CD8 cells, we're going to ultimately make both memory TH cells or helper T cells and active TH cells or helper T cells that are, so the memory T cells are just going to hang out and wait for the next time, and the active TH cells will be utilized for this particular pathogen. Okay, so active helper T cells can participate in the preparation of the cytotoxic T cells, as we mentioned. And then also they can stimulate B cell activation, which we're going to talk about next. And one thing that's important to note, just sort of to keep in mind, is that if the TH or the CD4 cells are destroyed, then all of adaptive immunity is impaired. And the primary example of CD4 cell destruction is with HIV. So it's a very real possibility, and the consequences are pretty significant for all of adaptive immunity. All right, so next let's consider the humoral immunity, which is also called antibody-mediated immunity. And like the T cells, we'll just consider the preparation of B cells. So B cells also come from hematopoietic stem cells, which ultimately become lymphoid stem cells and then B cells. And the B cells mature in the bone marrow. So unlike the situation with the T cells where they go to the thymus, they stay in the bone marrow until they're mature. Once the B cells are produced, they will ultimately be transferred to the secondary lymphoid organs, the spleen, the lymph nodes, etc., just like the T cells, where they're going to wait and encounter some antigens. So, one thing I want to just mention before when we talked about the APCs, we drew a phagocyte for the class 2 MHC, and we discussed a mechanism for how we would have entry of a pathogen into the cell, which ultimately could be broken down into pieces and then a fragment could be displayed. In the case of B cells, it's a little bit different. We're actually going to have we're going to have the antigen bind to antibodies that are present on the B cell. So the antigen binds to an antibody, it is then engulfed, and then once it's engulfed, the process continues as we described before, where the foreign antigen fragment will display on the class 2 MHC. And this process is referred to as B cell sensitization. And once B cell sensitization is complete, we're going to have interaction with the helper T cells. So as we mentioned before, the helper T cells participate in B cell activation, and that's what we're going to have next. So we have our original cell here that is displaying the antigen, and then we also have our helper T cell that's come by to take notice of this antigen that's problematic. And the co-stimulation is going to happen here is going to include release of cytokines. So remember, those are small protein chemical messengers, and the release of these cytokines is going to result in B cell activation. So the B cell is going to divide into memory B cells and activated B cells. Activated B cells will divide into plasma cells, or differentiate into plasma cells, and then the plasma cells will secrete antibodies. And in the next video, we're going to talk a little bit about what the specific functions and purpose of these antibodies is. So that concludes our discussion on cell and humoral immunity, just the basic mechanisms that can deal with antigens and problems in the body. So I hope you enjoyed it. Thanks for watching.